In this multiple choice question from the given option, they are directing us to specify the correct vector option used for cloning the small DNA fragment. For this, we are ignoring the wrong option to choose the correct option. Why we are choosing plasmid? Because it is in a position to accommodate the small DNA fragment without corrupting all the regulatory gene, so that it can bring the effective multiplication of the small DNA fragment at a fast rate, unlike bacterial artificial chromosome, yeast artificial chromosome, and plasmid. So we are choosing option C as a correct answer to secure 4 mark. In this pictorial representation about the plasmid vector, we are specifying after introduction of the given gene into the plasmid by using DNA ligase enzyme and restriction endonuclease enzyme. We are making this recombinant plasmid to get multiplied into multiple copies, so that the given gene is also getting multiplied into multiple copies, which is shown as a red color in this blue color plasmid circle. This plasmid is responsible for cloning the given DNA into multiple copies. So we are choosing option C as a correct answer to secure 4 mark. What are the points we told with this pictorial representation? We are giving as a point-wise narration to take the NEET notes. Small circular DNA molecule or plasmid or extra nucleo and carry extra chromosomal gene found in certain fungus and bacteria. They produce on their own the best known vector which are also available commercially are PBR 322 or Bolivar and Rodriguez plasmid and PUC18 vector. So with this specification, we are choosing option C as the correct answer to secure 4 mark.